Good morning, dear students. This is your teacher Neela Mehlawat, and we will today complete our chapter 8, the cell, the unit of life. In the previous video, students, we have talked about the various cell organelles that are present in the eukaryotic organisms. And in that continuation, we'll talk about the last, but not the least, that is your nucleus. I hope everyone has heard this word nucleus. Have you heard, everyone? So can you tell me what is a nucleus? Okay, yes, tell. Yes, exactly. See, in a cell there are three things, whether it is a plant cell or an animal cell. All types of cells, they generally contain three things. One is a plasma membrane. Second is, in the center there will be a nucleus, a darkly stained body which will be present, that is called as nucleus. And then there is a cytoplasm. So three structures, they are very common to all these cells: the plasma membrane, cytoplasm, and the nucleus. Now, have you wondered who discovered this nucleus? Now, Robert Brown. I hope you've heard this name. Robert Brown is a scientist who described the nucleus in 1831. So the onus of discovering the nucleus goes to Robert Brown. Now, along with this, there was another scientist named as Fleming. He also identified that the material of the nucleus, when it is stained by basic dyes, okay, the substance which is present inside the nucleus, it gets stained with the basic dyes, and thus it was given a name which is called as chromatin. So we can say that inside the nucleus the chromatin is present. And normally we will observe that in most of the cells there is only one nucleus. You can see here also, this is a cell, this is a plant cell. So you will find generally a single nucleus is present in a cell. But some cells may have more than one nucleus. Like your neutrophils, they can have more than one nucleus. While in some cases you will find that the mature cells, they do not contain nucleus. Like your RBC, the mature RBC, it does not have a nucleus because it is filled with hemoglobin. And in the same manner, in plants also, the sieve tube cells, the cells which are present in the sieve tube, they also do not contain the nucleus. So we observe variation in the presence of a nucleus. Some contain one nucleus, some contains more than one nucleus, and we also observe some examples where even the nucleus is completely absent, like in case of red blood cells and the sieve tube cells. Now, when we observe the cell in a non-dividing phase, or you can say in a resting phase, so resting phase is also called as interphase. Okay, when we observe the cell when it is in resting phase or when it is in interphase, then it contains the four components. One is the nuclear envelope, second is the nucleoplasm. Like in a cell you have a cytoplasm, so in the nucleus you have a nucleoplasm. The another name for the nucleoplasm is nuclear matrix, chromatin, and the nucleolus. Okay, nucleolus is a singular word, and nucleoli is plural word for this. So the electron microscopy, now just focus, let's focus on the diagram itself. This complete is your nucleus. Okay, in the cell there is a nucleus, so this is your nuclear envelope. Then there is a chromatin which is present in the nucleoplasm and nucleolus. So three structures. Now let's start with the nuclear envelope. Now let me magnify this diagram. See, nucleus is surrounded by double membrane, Okay, the nucleus, it is surrounded with two membranes, or we can say it is surrounded by a double membrane. The space between the two membranes, as you can see, this is the outer membrane, this is the inner membrane, and there is a space between these two membranes. This is called a perinuclear space. So we have two membranes, outer membrane and the inner membrane. Then in between these nuclear envelope, there is a space which is called the perinuclear space. Now this perinuclear space, it forms a barrier between the materials that are present inside the nucleus and that of the cytoplasm. तो materials जो हैं वो इसको cross करके नहीं जा सकते हैं. Okay, the substances which are present in the nucleoplasm, they cannot go through this double membrane structure in the cytoplasm, or the materials which are present in the cytoplasm, they cannot enter into the nucleoplasm. But we know that nucleus always brings about some kind of transport. So you will observe at these places, just look at these structures. These are referred to as nuclear pores. So at a number of places, the nuclear envelope, it is perforated by minute pores, and these pores, okay, these pores, they are formed by fusion of these two membranes, and these are the pores through which these substances may go from the nucleus into the cytoplasm, or they can come from cytoplasm into the nucleus. So nuclear envelope is perforated by a number of nuclear pores, and through these nuclear pores, the transportation of RNA, the transportation of protein molecules can occur in both the directions. Okay, so there is double membrane structure. The space between the two membranes is called as perinuclear space, and it does not allow the materials to pass through the membranes. So at specific distances, you will find certain pores which are called as nuclear pores, and these nuclear pores, they help in transport of the materials between the nucleus and the cytoplasm. Now coming to the second portion, the new chromatin part. See, in the nucleoplasm, can you see these thread-like structures? So in the nucleoplasm, there is a network of nucleoprotein fibers. जो न्यूक्लियोप्लाज्म है न्यूक्लियस की, उसके अंदर एक नेटवर्क प्रेजेंट है फाइबर्स का, न्यूक्लियोप्रोटीन फाइबर्स का, and these nucleoprotein fibers are referred to as chromatin. Okay, so during resting phase, when the cell is not dividing, so you will observe a network of fibers, or you will observe chromatin. But when the cell passes during the different stages of cell division, this chromatin material, it condenses to form the chromosomes. ओके, जब आपका सेल जो है वो डिवाइड करना शुरू करेगा, उससे पहले ये क्रोमैटिन मटेरियल जो है वो कंडेंस होकर क्रोमोसोम्स बनाएगा. ओके, एंड दिस क्रोमैटिन मटेरियल, इट बेसिकली कंटेन्स डीएनए. जो आपका एक क्रोमैटिन मटेरियल है, इसके अंदर डीएनए प्रेजेंट होता है. इट कंटेन्स सम बेसिक प्रोटीन्स, एंड दोज बेसिक प्रोटीन्स आर कॉल्ड एज हिस्टोन्स. And in addition to this, there are some non-histone proteins and RNA. So the chromatin basically consists of four things: DNA, histone proteins, non-histone proteins, and the RNA. And when the cell will divide, okay, when the cell becomes ready for the cell division, then this chromatin material, it will condense to form the hard structures that is called the chromosomes. Okay, so chromatin will form the chromosomes, right? Now, apart from the chromatin material, the third structure which is there is the nucleolus. So in the center of the nucleus, there is a darkly stained body. Okay, in the center of the nucleus there is a darkly stained body, and this darkly stained body is called the nucleolus. What do we call it as? It is called the nucleolus. It is basically a spherical structure. Okay, it is basically a spherical structure which is present in the nucleoplasm. Okay, and it is not surrounded by a membrane. Nucleus, we know it is surrounded by a nuclear envelope, but nucleoli or nucleolus, it is not surrounded by any membrane. And we can observe that the contents of the nucleolus, they are in continuation with the nucleoplasm. So your nucleolus, it remains suspended into the nucleoplasm, and it is a place where ribosomal RNA synthesis occurs. So it is a site where ribosomal RNA synthesis occurs. So in some cases you will observe that there are more than one nucleoli. So if a nucleus contains more than one nucleolus, then in that case the synthesis of proteins, the rate of synthesis of proteins will be higher. If a nucleus contains only one nucleus, then the rate of synthesis of protein will be less in comparison to the nucleus which contains more than one nucleolus. So as I told you, the chromatin, it will condense to form the chromosomes. So in a human cell approximately there is 2 meter long DNA is present. अगर सेल रेस्टिंग फेस में है तो उसके अंदर जो भी DNA मटीरियल है वो फाइबर्स की फॉर्म में होगा, एंड देन इन दैट टाइम वी विल कॉल इट एज क्रोमैटिन. सो जो क्रोमैटिन की लंबाई है वो अप्रॉक्सीमेटली 2 मीटर लंबी होगी. ओके, इट इज अप्रॉक्सीमेटली 2 meters long. Now when the cell prepares itself for the cell division, then this 2 meter long chromatin fibers, they will arrange themselves to form the 46 chromosomes or the 23 pair of chromosomes. So this 2 meter long chromatin material will condense to form the chromosomes. Now one chromosome, see, you look at the structure of a chromosome. A chromosome basically is made up of 2 equal parts. And those 2 equal parts, they are called as chromatid. What do we call them as? Chromatid. So this is one chromatid. This is one chromatid. This is second chromatid. So 2 chromatids together form a chromosome. Okay, and they are joined from the center they are joined by a primary constriction. Okay, they are joined by a primary constriction or the centromere on both the sides. Okay, and on this you can say centromere on the sides of which a disc shaped structures are present. तो सेंटर में जो स्पेस है वो आपकी सेंट्रोमियर है, एंड एक डिस्क शेप स्ट्रक्चर प्रेजेंट होता है सेंट्रोमियर पर जिसको हम कहते हैं काइनेटोकोर. सो दिस काइनेटोकोर, it connects these 2 chromatids and form a single chromosome. So centromere, it holds the 2 chromatids and it forms one chromosome. Okay, and the disc shaped structure on the centromere is called as kinetochore. Now depending upon the position of the centromere, okay, depending upon the position of the centromere, we can have 4 different types of chromosomes. Okay, let's focus on this diagram itself. If the centromere is present in the center, okay, if the centromere, it is present exactly in the center of the chromosome, and both the arms, this is one arm, this is second arm, both the arms, they are of equal size. So such a chromosome is called as metacentric chromosome. What do we call it as? Metacentric chromosome. Okay, centromere is exactly present in the middle and there are 2 equal arms of the chromosome. So chromosome के दोनों हिस्से बराबर होंगे, आपका centromere exactly center में होगा. So such a chromosome will be called as metacentric chromosome. Second, in the second type, the centromere, it move towards the one side. So one arm will become longer, another arm will become a slightly shorter than the longer arm. ठीक है, अब आपका जो सेंट्रोमियर है वो स्लाइटली मिडल से थोड़ा सा दूर हो गया है, जिसकी वजह से एक आर्म थोड़ी छोटी हो गई है और एक आर्म थोड़ी सी बड़ी हो गई है. सो सच अ क्रोमोसोम इज कॉल्ड एज सब मेटासेंट्रिक क्रोमोसोम. सब मेटासेंट्रिक क्रोमोसोम. Metacentric chromosome, when the centromere is present in the center and both arms are equal. Submetacentric, when the centromere move a little away from the middle, so one arm becomes a little shorter, another becomes a little longer. Third, again the centromere move towards one end of the chromosome. So in this case, one arm becomes longer than the another. So one is extremely short, another is extremely long. एक आर्म जो है वो ज़्यादा ही बड़ी हो जाती है, एक आर्म है जो काफ़ी छोटी हो जाती है. So such a chromosome is called as acrocentric chromosome. What do we call it as? Acrocentric chromosome. Now last one is a telocentric chromosome. In case of telocentric chromosome, the centromere moves to one terminal. तो ये जो आपका क्रोमोसोम है जिसे हम कहते हैं टीलोसेंट्रिक क्रोमोसोम, टीलोसेंट्रिक क्रोमोसोम में जो आपका सेंट्रोमियर है वो ऑलमोस्ट एक टर्मिनल पोर्शन पर पहुंच जाता है. ठीक है. सो इट हैज अ वेरी वेरी शॉर्ट आर्म एंड अ वेरी लॉन्ग आर्म. So this kind of chromosome is referred to as the telocentric chromosome. So in totality we have 4 types of chromosome: metacentric chromosome, submetacentric chromosome, acrocentric chromosome and the telocentric chromosome. Now in addition to this, in some organisms or in some chromosomes there are some non-staining secondary constructions are present. Just look at this chromosome. At one end of this chromosome there is a secondary construction and there is a secondary construction and there is a small fragment, and this small fragment is called satellite DNA. तो ये छोटा सा फ्रेगमेंट जो आपको नजर आ रहा है ये क्रोमोसोम के ऊपर एक सैटेलाइट डीएनए की शेप ले लेता है. Okay, so we can have a satellite also. So 4 types of chromosomes: metacentric, submetacentric, acrocentric and telocentric chromosomes. While some chromosomes they also have non-staining secondary constructions, and these non-staining secondary constructions they are called as satellites. Now coming to the last portion, that is microbodies. Okay, now if you look at the diagram, माइक्रोबॉडीज जो हैं ये एक वेसिकल्स हैं जिनके चारों तरफ एक बायोलिपिड लेयर है, एक प्लाज्मा मेम्ब्रेन भी है, एंड इन द सेंटर देयर इज अ क्रिस्टलाइन कोर आल्सो. नाउ दीज माइक्रोबॉडीज दे आर माइन्यूट वेसिकल्स. Okay, these are minute vesicles and they contain their various enzymes. ये छोटी छोटी वेसिकल्स हैं, मेम्ब्रेन बाउंड वेसिकल्स होती हैं, इनके अंदर अपने एंजाइम्स प्रेजेंट होते हैं और ये दोनों में प्रेजेंट होते हैं. These are present in both plants as well as in animals. Okay, they are present in animals as well as plants. So we can say these are enzyme filled vesicles which are present in plants and animals. Okay everyone, I hope this much is clear to everyone. Okay, so to sum up, I have to differentiate between the 2 types of cells, that is the plant cell and animal cell. So in case of plant cells, cell wall is present. In animal cell there is no cell wall. So here you have plastids are present, plastids are absent. In plants there is a central vacuole. In animals the vacuoles they are large in number but they are small in size. Plants, the centrioles are absent, but in case of animals the centrioles are present and they help in cell division. Now in addition to this, let's discuss the prokaryotic and eukaryotic cells also. Prokaryotic cells, they are generally smaller in size. Okay, they are generally smaller in size, while your eukaryotic cells, they are larger in size. The genetic material is observed in the form of nucleoid, or we can say nucleus is not well developed. Nucleus is well developed. Nuclear membrane is absent, but here nuclear membrane is present. Various membrane bound cell organelles they are absent. The various cell organelles they are present. Here the chromosome, it is in the form of a circular DNA. Here the chromosome is in the form of a linear DNA. The ribosomes that are observed in case of prokaryotic cells, it is of 70S type, while in case of eukaryote it is of 80S type, except the plastids and the mitochondria, they contain 70S type ribosomes. So I hope dear students, you've understood the all concepts of this chapter, because with this topic we have finished the chapter also. So if there is any doubt, you can ask me. Okay, so today's your homework is you will make notes of this topic. You will read your page numbers from 138 to 140. Right, you will read the page numbers from 138 to 140, and you will underline the important points. You will try to frame the 1-mark questions, and especially focus on those words which are there highlighted with black. And if there is any doubt, any query that you feel, you can discuss with me. You can send your queries to me. Okay, so that's all for today. Bye everyone.